Hi Year 11, welcome to our next video on enzymes. Before you start, I hope you've watched the video from the Amoeba Sisters enzymes and made your own notes so that you're familiar with how enzymes function. In this lesson, we're going to be focusing on the effect of environment on enzyme activity. So what I want you to know, your takeaway, is that enzyme activity can be affected by a variety of factors such as temperature, pH, and substrate concentration. Enzymes work best within specific temperature and pH ranges, and suboptimal conditions can cause an enzyme to lose its ability to bind to a substrate and therefore not work efficiently. Let's first of all look at the effect of temperature on enzymes. Due to their protein nature, enzymes are sensitive to temperature. Enzymes within cells function best at body temperatures. Above 40 degrees Celsius, their rate of reaction, their efficiency decreases, and at 60 degrees Celsius, most enzymes stop functioning altogether. This is known as denaturing. So temperature affects all proteins. Because enzymes are also a class of protein, they're also affected by changes in temperature. So if you heat an enzyme above the temperature at which it works best, above its optimum temperature, they stop working. If you cool enzymes down, their activity slows down until they eventually no longer work. But if you reheat the enzyme, it will once again work until it reaches its optimum temperature where it's most efficient. So each enzyme will have an optimal temperature where it works most efficiently. Once you go past that optimal temperature, okay, it's too hot, then the enzyme is denatured or damaged. So when it's denatured, it means its shape no longer fits the substrate. So the enzyme can no longer work. The shape change caused by heating damages the enzyme molecule permanently, whereas cooling the enzyme does not cause permanent damage. So here we have a nice graph modelling what is actually happening here. So our environmental factor is temperature along the x-axis and rate of reaction along the y-axis. So what we have here is some enzyme. We can see starting from 0 degrees Celsius, as you increase the temperature, the rate of reaction increases. Why is that? Because heat energy causes more collisions between the enzyme and the substrate until we reach that optimal temperature. Okay, and for a lot of human enzymes, that'll be around 37 degrees Celsius. Okay, past that, you can see the rate of reaction rapidly decreasing down to 0. Why is that so? Because heat actually damages the protein structure, so the active site changes, it can no longer bind to the substrate, and so that reaction cannot occur. So just a little bit more about denaturing. Heat causes the hydrogen bonds that maintain the form of the enzyme to break and therefore alters its structure and shape. When that happens, we say the molecule or the enzyme has been denatured. The change in the shape affects the active site, and so therefore the substrate cannot bind. Extreme cold can also slow down an enzyme's function. So here we have an image that's actually modelling what is happening there when an enzyme is denatured. So here's our enzyme, and we have our active site here. You can see that it's going to fit nicely with the substrate. But what happens if you heat it above 40 degrees Celsius, which I assume it's optimal temperature? You can see the shape of the enzyme has changed. So bonds have been broken, the shape has actually been changed. The active site of the enzyme changes shape and it can no longer bind to the substrate. When that happens, we say it has been denatured. Another environmental factor is pH. So proteins are sensitive to changes in pH. Enzymes are a class of protein. Each enzyme has its own narrow range of pH within which it functions most efficiently. Levels of alkalinity or acidity outside an enzyme's optimum pH alter the shape of an enzyme's active site, stops its functioning, causes it to denature. Now, a lot of the enzymes found in cells function in a neutral pH. Not always the case though. Pepsin and renin, found in the stomach, function best at a low pH. Salivary amylase functions best at a weak alkaline pH. We're going to be conducting practical activities to look at the effect of pH on enzymes, so you can also see Surfing Worksheet 72, Enzyme Activity and pH. Now here we have a graph showing the effect of pH on enzyme activity. So we have pH units along the x-axis. We have relative activity on the y-axis. So we have these pepsin and trypsin are two proteases. So prot for protein, A's for enzyme. So these are enzymes that relate to proteins. So here we have pepsin. We know now that it's found in the stomach, and you can see here its optimum pH is around 3, according to this graph. Here we have trypsin, okay, and you can see that trypsin's optimal pH is actually slightly alkaline. Not surprising, since trypsin as an enzyme functions in your small intestine. Finally, the effect of substrate concentration. So enzymes will work best if there's plenty of substrate. As the concentration of substrate increases, so does the rate of enzyme activity. However, the rate of enzyme activity doesn't just keep going up and up and up and up and up, because remember, there's going to be a point reached when enzymes become saturated and no more substrates can fit. So the enzyme has to be available to bind to the substrate to catalyze that reaction, but there's only a certain amount of enzyme molecules. Once they're all used up, once they're all bound to substrate, you can't speed up the reaction anymore. So here we have a graph showing substrate concentration increasing on the x-axis and rate of enzyme activity. So you see, as the substrate concentration increases, the rate of enzyme activity increases until here. Now, when you look at this, does this mean that enzyme activity has stopped? No, because okay, stopped would be down here, 0. What it means is the enzyme activity is continuing, but it's not going up any further. This is our maximum rate of enzyme activity. So our takeaway message, as the substrate concentration increases, so does the rate of enzyme activity until the optimum rate is reached at the enzyme's optimum substrate concentration. So even if you continue increasing the substrate concentration along here, okay, you get the same enzyme activity because there's not enough enzyme molecules available to break down the excess substrate molecules. So I'm going to actually ask you to have a go at the Titan worksheet, Factors Affecting Enzyme Activity. Okay, before you start that, have a look at these key points. Okay, remember that enzymes are a special class of proteins that speed up reaction rates. Without them, metabolic reactions would not happen quickly enough to meet the needs of cells. They operate best under optimal conditions. They need their optimum pH and their optimum temperature. Outside of those, the enzyme will not work as efficiently, which means the rate of metabolic reaction will be less, and in extreme conditions, the enzyme can even denature. Substrate concentration is the amount of substrate molecules available to combine with the enzyme. The higher the substrate concentration, the faster the rate of reaction until saturation of the enzymes is reached, and then it plateaus. So the reaction keeps going, but at its fastest rate, that's it. It doesn't go any faster. Some enzymes require the presence of a coenzyme, another vitamin or mineral ion, to help the enzyme-mediated reaction reach those optimal rates. If you need to, on the following slides, there are some helpful graphs. Okay, for example, this one that you can actually help check your answers. Okay, so you understand what you should actually be looking at. Once we finish this lesson, we're actually going to start our practical activities, investigating in a hands-on way the effect of pH and the effect of temperature on enzyme reaction rate.